let us look at some examples of antifreeze proteins with known structures. So we have seen four types of antifreeze proteins, 1, 2, 3, 4, and antifreeze glycoproteins. These are present in fish, and here are the structural or the polypeptide features. They have the alpha helix with the alanine rich, means they belong to type 1. If they have alpha, beta and loop structures with a C-type lectin, means they belong to type 2. If they have a globular cluster or short beta strands, they belong to type 3. If they have a helix bundle, they belong to type 4. And antifreeze glycoproteins have glyco groups. And in the case of insects, we have the beta helix right-handed and beta helix left-handed present in two different species. And when we look at the microbe, basically the bacteria, they have the globulin like a beta twist calcium 2+ binding domains. Fungi has a dimeric beta helix and right-handed structure. Plants, we have two major types. One is angiosperms. They have the beta roll domain with 8 loops of 14 to 15 amino acids. That's the theoretical one people had studied. And the diatoms have 1 alpha helix and 7 beta loops. So here are the structures which we can see. The molecular weight of each protein also varies. In the case of antifreeze glycoprotein, they have AAT repeat, that is alanine, alanine, threonine group is repeated, and they have the disaccharide unit as shown here is also present. Here is the threonine, alanine, alanine structures, multiple times they are present. The natural sources include Antarctic notothenoids and northern cards. And when we look at the AFP1, they have the molecular weight around 3.3 to 4.5 kilodaltons. They have alanine rich amphipathic alpha helical polypeptides as shown here. They some of the examples include right-eyed Flanders and baffin flies. And when we look at the type 2, the molecular weight is around 11 to 24 kilodalton. They have a disulfide bond, and some examples are given which contain sea ravens, smelt and herrings have this particular type of protein. When we look at type 3, the molecular weight is around 6.5 kilodalton. They have twisted loops folded into triple stranded beta sheets as we can see here. And ocean sprout, wolfish and eel pout are examples of species which contain these proteins. And when we look at type 4, the molecular weight is around 12 kilodaltons. They are alanine rich and they have a helical bundle structure as shown here. And here are the various general functions of antifreeze proteins. Their primary role is to protect organism from freezing by increasing freezing tolerance through ice adhesion. Some of the structural variation include they have a diverse three-dimensional structures with various motifs for binding. And how do they bind? The general AFPs bind irreversibly to the ice surface to inhibit its growth. In the later part, we will be studying about the mechanism also, how they inhibit the growth of ice. And the structure varies from organisms, and they also affect the efficiency of ice binding by the species. We have different types of structures which include long alpha helix or helix bundles, beta solenoid, etc. The molecular weight varied from 3.2 to even 1.5 megatons in Arctic bacteria. So the evolutionary pressure was under harsh conditions. This was actually produced. Across various diverse species, that is northern and southern hemisphere fishes have different types of AFPs. When we look at the gene origin, some of the AFPs are derived from genes like trypsinogen and chitinases. The common motif found is beta solenoid motif, which is prevalent in AFPs from bacteria, fungi, insect and plants. The adaptation mechanisms include evolutionary diversity allows for adaptations in cold environment, enhancing survival strategies. And all these things have different structural variations. And here are some of the structures of antifreeze protein which is given. The first one is antifreeze glycoprotein. They have the disaccharide linkage as shown here. We have the alanine, alanine, threonine structures. Again, alanine, alanine structures are given. Similarly, antifreeze glycoprotein with proline is also another type where proline moiety is present after alanine, alanine, threonine structures. So either alanine or proline may be present in this one. That means the proline is replaced by an alanine group. And the next one is antifreeze glycoprotein with arginine. So this arginine is in place of threonine, that is loss of the disaccharide also is actually here we can see the disaccharide present in the threonine. So this disaccharide unit may also be lost, and the proline one will have a alpha configuration as shown here, and there is also gamma methyl group which is present, and the arginine unit is actually not having our glycoside linkage. So that is the major difference. So these are some of the different types of antifreeze proteins. Let us look at the ice crystal, which is a very important component in understanding how the ice crystal growth is being prevented by these compounds. We know the HOH bond angle is 104 degree 5 minutes, and the HO bond length is around 0.9 angstrom. The major component of the ice crystal lattice, which is written as 1H, is a water molecule. This is surrounded by 4 neighboring molecules and linked to them by hydrogen bonds. We will actually look at the structure soon. The liquid water has a 4.4 neighbors compared to 4 neighbors in the ice crystal 1H. So 4.4 neighbors gives higher density for water compared to the ice. That's the reason water is denser, ice is lighter, and ice floats on water. So here is the crystal structure of theoretically developed structure by density functional theory is shown here. Here we can see a nice cyclohexane chair-like conformation which has hydrogen and oxygen which are present here. So if you look at the red dots are basically the water molecules, the ash color ones are actually the hydrogen atoms. So there is lots of hydrogen bonding that is present in this one, and if we very carefully look at every oxygen has, in other words, one hydrogen is actually surrounded by 4 oxygen atoms. So that is how the structure actually goes here. So we can see in this one, this hydrogen atom has 1, 2, 3, 4. 4 oxygens are being surrounding one hydrogen atom. So this actually repeats throughout 3-dimensionally. So that is how the ice crystal structures are actually formed. And here we have Bernal and Floer ice rule, which talks about how these crystal structures are actually formed. So each oxygen is covalently bound to 2 hydrogen atoms, and that the oxygen atom in each water molecule forms 2 hydrogen bonds with other water molecules. So that is precisely 1 hydrogen atom between each pair of oxygen atoms. That means 4 oxygen atoms surround 1 hydrogen. So this is what it is given in the Bernal-Floer ice rules. So where is the place where these hydrogens actually occupy? So if a proton is positioned in the middle of a hydrogen bond, proton here means hydrogen is positioned in the middle of the hydrogen bond, it will result in a ionic crystal structure with a high melting point, which is much higher than 100 degrees centigrade, because we know ionic crystals will have higher melting point, but water is having a lower melting point of 100 degree only. And it also has huge electroconductivity of the liquid phase water and has a much smaller dielectric permittivity of epsilon equal to 10. Whereas in the case of ice, it is epsilon is equal to 100. So that means hydrogen is not actually present in the middle of the hydrogen bond. So then where is actually it is present? So Pauling ice rules tells about where the hydrogen may be present. So the distribution of bonds on this lattice is represented by a directed arrows and can be either ordered or disordered. So here we will be looking at how they are present. So the hydrogen atom can assume one of the 2 positions of the hydrogen bond, and hydrogen switches from one position to another many times per second. So proton and hydrogen are basically here used as synonyms in a simple way. There are 6 different arrangements where we can place the hydrogen atoms in the ice 1H crystal structure. So how they are moved is shown here. They move from in a clockwise direction as shown here in this particular case. They may also move in different directions. So here if you look at the hydrogen atoms, each hydrogen atom is surrounding an oxygen atom. So here this hydrogen is kept far from this particular oxygen atom. This hydrogen is kept far from this oxygen atom. This hydrogen is kept very far from the central oxygen atom. This hydrogen is kept far from this central oxygen atom. So that means one oxygen also has 4 hydrogen atoms. Similarly, one hydrogen atom also has 4 oxygen atoms. So that is how they are actually placed. So if you look at the other one, this hydrogen is actually placed very close to this oxygen. This hydrogen is placed very close to this oxygen. These two hydrogens are placed near these two oxygen atoms. So in other words, what we can say is hydrogen is not kept exactly in the middle between two oxygen atoms, but in some cases they are closer to one oxygen, they are longer from the other oxygen. Here we have 1, 2. These two hydrogen atoms are closer to the central oxygen. Other two hydrogens are closer to the outer oxygen. So this is how we can actually see how they are placed. But when we go to the second structure, we have this hydrogen atom which is far from the terminal oxygen, but it is close to the central oxygen atom. So if we look at the 3, 1, 2, 3, now there are 3 hydrogen atoms which is close to the central oxygen, whereas one hydrogen which is far from that oxygen. But in the other way, if you look at 1, 2, 3, 3 hydrogens are kept far away from the terminal oxygens, whereas one hydrogen is kept close to the one of the terminal oxygen. So that way we can say, if we assume that it is a tetrahedral structure, of course in crystal it is a continuous structure, so the positions will keep on changing. So these are all the various 6 types of arrangements which can be made according to Pauling's ice rules.